Yeah.
Yeah. Okay. So you need to remember that we have these very peculiar kind of cells called as the white blood cells that are going to help us in the process of gathering immunity against any particular kind of disease that may try to attack us. Okay. Now, when I say that we are going to have white blood cells, in this particular chapter, we need to remember about two important white blood cells. The first one that are going to have the function of eating up your microorganisms. So let's say there is a particular pathogen, okay, that is going to come and try to attack you. These macrophages are going to be slightly bigger cells. They are going to have a nucleus, okay, and along with the nucleus, they're going to have a lot of digestive material that is going to be present in their cytoplasm. What these macrophages do is they spot the pathogen. They identify the pathogen as a non-self structure. Upon spotting and identifying a particular pathogen as a non-self structure, they go on to neutralize that particular pathogen. Now, how do they do this neutralization is very important. So in order to neutralize these particular pathogens, okay, what they are going to do is they are going to engulf this pathogen. How do they engulf this pathogen? So they are going to quickly move towards this pathogen, okay? And once they go toward this pathogen, they are going to take that pathogen inside their body through a process called as endocytosis. Okay, and by endocytosis, what they're going to do is they're going to take this pathogen all the way inside their body, and then slowly they will break this pathogen into smaller and smaller pieces, and then they will be killing this pathogen. Okay, so that's basically what happens in your process where the macrophage cells, these ones, go on to eat the pathogen. When this is done, okay, this process is called as macrophagy or phagy, however you want to call it. And
Why not phagocytosis?
Sorry?
Why not phagocytosis?
You can call it phagocytosis also. So phage, okay, the word phage means this particular pathogen. So when you eat this cell, so macrophagy is one term, but it can also be known as your phagocytosis, which means that a cell, cyto is a cell, eats this phage or the pathogen. So phagocytosis is nothing but the same process, and in MCQs, you could also be given another term for it, which is macrophagy. Okay? That makes sense?
Yeah.
Yeah. Okay. So now these are your primary cells of importance when it comes to your immune system. Along with this, now when we said that you need to identify these cells as non-self, okay, this is a very important concept for your body, which is identifying something as non-self. What it means is all of the cells of your body have a particular tagger on them. Okay, so what this tag does is it allows your body to identify that, okay, because this tag matches to all of the cells, these cells have to be coming from my body. Okay, so this is an identification tag that these cells have. In conditions where this tag is missing from your cellular structures, your body goes on to identify it as non-self, which means it essentially just tells your body that this particular cell cannot be from your own cellular origin. It has to have come from outside. This kind of response of understanding something as non-self also occurs in human bodies when you are pregnant. So your body identifies a embryo, okay, or the growing baby for a period of about 3 months as non-self. And that is the reason why you may have noticed that the pregnant women have different sort of symptoms where they have an excess amount of vomiting, or they have a lot of pain in their stomach, or sometimes, if you watch those Hindi serials and things, you will see they sort of show that the women sort of faint a lot. Okay, all of these symptoms are because the body doesn't identify that particular cellular structure as self. Now, once you identify something as non-self, okay, what happens is there are certain cells which are called as your neutrophils. Okay, what these neutrophils are going to do is they help in or just not reciprocating, just tagging these cells as non-self. So once if they have been identified as non-self, a certain kind of tag gets added onto these cells, and then these cells are going to be taken to your immune system. So I'm just going to write this over here. These cells will be taken to your immune system, and these cells will be presented to your immune system so that they can be killed. Okay, so when you take a cell and present it to your immune system, you will be called as presenter cell. What are you doing? Your job is to take the pathogen and present it to your immune system. Your neutrophils, okay, are going to do that function, and therefore they're also called as the presenter cells. That is the only term that we associate with them. Okay, so I'm just going to write this over here. Presenter cells. So this is like a basic introduction to all these different kinds of cells. Okay, I'll take you in the detail of this part as well. Once you understand that you are going to have this process of phagocytosis take place, okay, there is one more very important term that you want to understand, which is when we just simply talk about your cells. So let's say this is a normal eukaryotic cell. This is very important that you remember. All of this immune response that you're learning about is taking place in eukaryotic cells. Okay, your prokaryotic cells do not raise an immune response like this. In fact, what they do is they would simply kill the entire cell so that the infection doesn't spread to the entire population, because your prokaryotic cells are mostly unicellular, right? So what they choose to do is they just choose to kill the entire cell. And when that is happening, just like your eukaryotic cell, they also have a structure that gets activated in their bodies, okay, or in their cells, which is called as a lysosome. What does a lysosome do? Lysosome contains lytic enzymes. What are lytic enzymes? Lytic enzymes are the digestive enzymes. So I'm going to write over here, digestive enzymes that are going to be packed into sac-like structures, okay? And whenever a cell receives a response that it is being threatened, or there are certain structures that need to be killed, the lysosomes burst open and the lytic enzymes come out and they kill all of the structures that come in their vicinity. Okay, that is basically what needs to happen. But before this entire thing can happen, now let's look at this in a form of a story. Okay, so let's say that this is a group of cells that are present over here. Once this group of cells has been identified by your body as self cells, they are not bothered at all. Your body doesn't really do anything to harm these cells. But there is a non-self cell that gets detected. As soon as this cell gets detected by your body, there are going to be certain cells called as the presenter cells, whose job is to take these non-self cells to your immune system so that they can be killed. But at times, what happens is, instead of taking this huge cell to your immune system, okay, because it could be heavy, or maybe it's a large number of cells that have attacked, or maybe it's just not possible for the presenter cells to take these non-self cells to a site where they can be destroyed. in that condition, what happens is you raise a immune response which works sort of like making a call to your immune system. Okay, so what you do is you send a chemical signal which works like a call. So I'm going to say chemical call, okay, which tells a certain number of immune response cells to come to the site of infection and then kill the non-self cells. This chemical call or this chemical attraction, because of which there is a recognition of these cells, and upon recognition, the process of endocytosis will take place. This chemical call is called as chemotaxis. Chemotaxis. Okay. What happens in chemotaxis is you're going to send a chemical signal or a chemical call to your immune system, and then these immune molecules are going to come to that spot and they are going to kill the pathogens. This is the only portion of the immune system that is different for human beings. Rest of the things will happen in all of the living organisms. Chemotaxis is very, very specific in human beings, and there are various kinds of chemicals that are going to be sent as a response. That means for every single pathogen, the chemotaxis is going to be done using a different kind of chemical. And that is how your body is able to differentiate as to for what is this chemical call being sent. Okay, so that's basically how it is going to work. Now let's see if you have a pathogen right over here. Okay, this pathogen has recently identified a particular bacteria. So what this particular cell is going to do, it is going to go and be near this bacteria, okay, so that it can take these bacteria and present them to your immune system. And then it realizes, oh, these are too many bacteria and I cannot take these bacteria to my immune system. So it will raise a chemical signal. So I'm going to write over here, chemotaxis will take place. Upon the proper initiation of the chemotaxis, what this cell will do is it will think, by the time my immune system starts to respond to something, what if these pathogens run away? You don't want that to happen. So it will release certain molecules, okay, which will be tagged onto this pathogen like this. And so what you do is you mark these cells in a particular manner so that your body can identify them. So you have these neutrophil cells. I'm just going to write the name over here. Okay, this is your neutrophil cell. And what has the neutrophil cell done? First, it has raised the chemical signal to let your body know that there are certain pathogens, such as bacteria, that need to be eliminated. Along with raising a chemical signal, it has also attached certain markers, okay, so that the bacterium can be very easily identified. This marker that you attach, this is a type of antibody. Okay, so I will just write over here a antibody marker allows for the easy identification of the pathogen, in this case, the bacteria. Okay, now once this cell has been identified because of the antibody marker that exists, the process of endocytosis will take place. The cell will be engulfed by your macrophage, and then upon being engulfed, the lysosome, the cell that we just spoke about over here, okay, this is going to break open and the lytic enzymes are going to kill that particular bacterium. Okay, now if the bacteria is going to be digested, all of these materials of the process of digestion of that bacteria are again going to be thrown out of the cell. So thrown out, and while taking in happened because of the process of endocytosis, throwing out takes place via the process of exocytosis. Similar way as the cell engulfed something, it also throws the substances out. Is that making sense? This is how you are going to have the phagocytosis take place, and the antibody is going to be attached to the pathogen. Now, at times, okay, I'm just going to write this over here. The pathogen can also be called by another name, which is antigen. Okay, so when this process takes place and you tag your antigen with this antibody, what you form is an antigen antibody complex. So I will just write this: antigen antibody complex. And it works exactly the same way that your enzyme and substrate complex works. So it basically goes to look something like this. So if this is your antibody, okay, each antibody is going to have a specific site to which the pathogen comes and attaches, and then this pathogen can be sent for the process of neutralization. Is that making sense? Did you understand till here? Do you want me to go over any part one more time?
No, yeah, I understood.
Yeah, this makes sense. Now,
Yeah.
this is how a particular antigen will be neutralized. Okay, now this seems easy and it makes sense that, okay, aise ho sakta hai. But this is for the cells that have a very straightforward mechanism of being identified. Okay, this will happen for those pathogens that did not try to obscure their identity. For the cells that just came to your body posing as pathogens, and your body was really easy and very capable of identifying these cells as non-cells. But there are certain pathogens that will come in your body and they will hide themselves with different markers on them. Okay, so let's say that your body has the cells that have a very specific kind of marker. Okay, let's say these are your liver cells. Okay, and your liver cells have a very specific tag to them, which allow your body to identify all the cells with this tag as your liver cells. Some pathogens can be very clever, and what they do is they follow a pattern where they also develop a similar kind of tag on them. And because of that, what happens is these pathogens will get inside your body and your body will never be able to identify that, oh, these cells are not your body cells. And therefore, they kind of get inside your body, start to grow, and they just multiply to a very large number. And after that, your body is able to identify that, acha, ye hai to liver cells, but what are they doing in my lungs? And then your body understands, oh, so I was just tricked, and these are actually not my liver cells, these are the antigens. And then the attack begins. Okay, but till that time, what has happened is your cells who attacked you have already grown in a large number. Now, because we don't want this to happen, you have different other kinds of cells that are always going to move around your body. Okay, they're just basically going to be like the patrol police. And what they do is they keep on looking for the cells that seem suspicious, like having liver cells in your lungs is suspicious. And although your entire body is saying that, yeah, that it seems suspicious that there are liver cells in your lung, but they're still our body cells, right? The lymphocytes will tell those cells that yes, they are your body cells. They look like your body cells, but we still need to kill them because they are in the wrong space. And those cells who have the smart sense in their body are called as lymphocytes. Okay, so lymphocytes are the cells that keep moving through body looking for pathogens. That is the only work that these cells keep on doing. The way to identify these lymphocytes under a microscope is you want to look for cells that look like phagocytes but are smaller than phagocytes. Smaller than phagocytes. I will show you a slide, okay, where you will be able to identify some of these cells. If you have your textbook in front of you, you can look at the image. I'll just tell you the number of that image. Just give me 1 second. Yeah, you can look at the figure 11.2. Okay, and in this figure 11.2, you are able to see certain cellular structures that are present. Okay, if you can spot, you will see that there are 3 very peculiar cells that are present. The one that is present on the right hand side, okay, on the extreme right, is a lymphocyte. It is smaller than the cell which is present on the left hand side, which is a phagocyte. Okay, so that's basically how you are going to be able to identify all of these cellular structures. It's going to be a little smaller than your monocyte or the larger phagocyte that is going to be present in your body. Now, what exactly is this lymphocyte going to do? What is the function of this particular cell? So this cell, along with having the ability to move through all of your body, okay, it is also going to be able to retain the memory of a particular kind of attack that might have taken place in your body. So these cells contain the memory of the attack. Okay, so this is supposed to be your brain, so that you remember that these are responsible for keeping a memory of all the different kinds of attacks from the pathogens that may have taken place in your body. Okay, that is a very specific work of your lymphocytes. Now, these lymphocytes are going to do this function, okay, because they can differentiate into 2 very important types of cells. One being your B lymphocyte and the other one being your T lymphocyte. I'm just going to write this B lymphocyte, and then you have your T lymphocyte. Okay, now once you see that you are going to have these B lymphocytes and T lymphocytes, it's also very important for you to understand that both these cells, B lymphocytes and T lymphocytes, so basically all the different kinds of lymphocytes, are going to be made in the same location where your RBCs are made, which is in your bone marrow. Okay, so when these cells are going to be created, a copy of these cells always exists in your bone marrow. It is there because of your genetic code. So if, let's say, you have been attacked or infected with a particular disease, the memory of that impact or that particular kind of attack gets stored in your body, and some of those cells will also go to your bone marrow, okay, and they will also pass on a certain amount of information there. And therefore, next time when you want to make more cells of the similar type, so let's say you got attacked by malaria in the first cycle, the second time if you get malaria, you will be cured by your body much more quicker than how long it took for your body to cure you in the first time. And this is possible because this memory gets sent to your bone marrow. It has the potential to make many, many more cells much quicker in the second time because your lymphocytes exist. Okay, this is also a reason why if there is an individual who is going to receive a bone marrow transplant from another individual, what we do usually is we do a lot of tests on this bone marrow, because if there are certain kinds of antibodies that are present in your system, okay, these antibodies could cause a interaction or could cause a rejection of the bone marrow when it is inserted into the body of another individual. I'll give you more clarity on this. Let's say you are a person who is going to donate the bone marrow to this individual B. Okay, you have been infected with malaria, you had malaria and you got cured, but B never had malaria. So their immune system is already weaker. That is why they require your bone marrow. On top of that, because you had malaria, there are antibodies of malaria in you. And when you give your bone marrow to B, along with having an entirely new bone marrow that is coming to their body, which is why their body is already going to be very vulnerable. On top of that, when they receive the antibodies for malaria, their body starts making a lot of antibodies against malaria. So even without having malaria, their body starts to react as if it does. And that can cause a lot of weakness. And in certain cases, that can also cause death of this individual or the recipient who is receiving this bone marrow. And therefore, it becomes very important for us to remember that while having memory of a particular disease is good for you, when you're donating your blood or you're donating your blood marrow, bone marrow, you have to be very cautious and you have to remember that you had this disease and you have to declare it to the doctor so that they can consider whether you are a good recipe or you are a good donor or not. Okay, so these informations are really, really important. So while having the memory is good, it can also cause a lot of dangers to your life if the memory is very strong. So at certain point of time, okay, which is something that is happening with people right now, which is because they have taken the vaccine. We are going to talk about vaccine also in this chapter, but I'm just going to give you a bit of intro here. So because certain people have already taken vaccine, there are antibodies against the COVID virus in them, right? Now, COVID virus is very similar in its structure and its abilities to cause diseases with a class of molecules. So corona is a class of molecules. There are many other viruses that look and behave similar to the COVID virus. Now, because of that, what happens is, if even a similar kind of virus infects their body, like something which is responsible for causing only common cold, their body thinks it is still the COVID virus and it reacts very vigorously to it. So where you are supposed to just fall ill for a day or two, your body raises such a huge amount of response that something that could have been cured in 2 days makes you so weak that you have to stay in bed for longer than 2 days. Okay, this is happening because people have already taken a lot of these antibodies in their body, and now their body remembers that, oh, this is the response that we have to raise. And because of that, it goes on to raise that very strong response to kill the COVID virus, and they sort of stay ill for a longer time. They have more fever, they have more body pain, they have a lot of weakness because of this memory that is present. So having memory is good, but having the memory of the same disease is also at times very difficult to deal with. Okay, so that's something you want to remember. I'll just put this in one corner here, and when we are talking about it, we can pick it up back. This we do not need. So coming to your T lymphocytes and B lymphocytes. Now these two types of cells that are going to be prepared by your body so that it can always make sure you're going to stay safe are really, really peculiar in what they do. So let's talk about your B lymphocytes first. Okay, B lymphocytes are the ones that are going to stay in your bone marrow. Okay, they are going to stay inside your bone marrow till they become mature. So unless and until they are released out, they are not going to be matured. So they are going to stay there as long as it takes for them to become older. And as soon as they become old, the first thing they start to do is just wander everywhere in your body. They don't have any specific direction where they want to go. They're going to go all throughout your body, but they follow a particular path to do that. Okay, so when they release out of your body, so I'm going to say first thing, mature in bone marrow. Once released, move rapidly through entire body. Another thing that you want to remember is they follow a pathway to go everywhere, which is they are always going to target your lymph nodes. Okay, now what are your lymph nodes? Lymph nodes are those structures in your system, okay, which are going to allow a lot of your body fluid to be reabsorbed if there are certain injuries, or if there's some extra tissue fluid that is present. All of that extra fluid in your body gets reabsorbed by these lymph nodes and gets added back to your circulatory system. So in your body, you are going to have multiple lymph nodes present everywhere. They are called as nodes because they have these structures somewhere in the middle that are going to be swollen like this. Okay, and these lymph nodes become extra swollen when you have different infections. So you will see your doctors will tell you that, yeah, you have swelling in your nodes, or you have swelling in your lymph nodes. That is an indicator of a very strong kind of infection. These B lymphocytes will increase in quantity in your lymph nodes when you have a stronger infection. That is what causes their extra swelling. Otherwise, they remain swollen. And if you feel excess amount of pain, or if you see that there is certain amount of inflammation in your body, what you can do is you can simply just massage these lymph nodes, and the excess amount of tissue buildup kind of gets distributed all over, and you start feeling better. But these B lymphocytes are going to use this particular structure, which is called as your lymph nodes, and they're going to move all across your body through these structures. Okay, along with your lymph nodes, your B lymphocytes also go to another organ, which is called as spleen. Okay, now what is spleen? Spleen is a structure in your body which is responsible for taking care of all of your dead RBCs. Okay, so there is a huge number of RBCs that die every day. Each RBC has the lifespan of about 120 days. After 120 days, each RBC dies off, and then it needs to be recycled. Certain amount of the material from an RBC gets recycled back, while some of it is used for making a pigment in your body, which is called as melanin. Melanin is the brown pigment that gives your skin a particular color. The Africans have excess amount of melanin, Asians have less amount of melanin, and your European people are going to have least amount of melanin. But that does not mean that they have lower number of RBCs dying. It just means that their spleen eradicates or cleans away the entire RBC, does not recycle any of it to make melanin. Okay, that is the basic function of your spleen. But along with removing all of these RBCs, your spleen also houses certain amount of B lymphocytes. What do they do over there is something we will discuss. But right now, just note that these are the basic points about your B lymphocytes. When it comes to the T lymphocytes, we usually do not say this entire word. That sounds to be really long word to use in daily communication. So instead of saying something like B lymphocyte and T lymphocyte, what we also do is we simply call them as B cells and T cells. Okay, there are no other cells that are called as B cells or T cells or D cells. We just refer those to these two structures. Now, what happens with these cells is they are also going to be present in the bone marrow till they mature. But once they are mature, they don't go everywhere in your body. They don't have this aimless life. They know exactly where they want to go. So I'm going to say leave bone marrow after maturing. Leave bone marrow after maturing and go to the thymus. Thymus is a butterfly shaped organ, okay, which is present in your chest region. So if this is your neck like this, okay, you will see that there are two bones that are present over here, which we refer to as collarbones. And then you have another bone like this to which your ribs get attached. The thymus is a yellowish organ which is going to be present right over here. Okay, this is what your thymus looks like, and this is where it is going to be present. Now, what does this thymus do? Thymus is a gland that is going to allow you to make certain kinds of hormones. Okay, and by the time you become an adult, the size of this thymus goes on shrinking down. So by the time you become an adult, if you started off with a thymus that was, let's say, this big, by the time you become an adult, it's going to be a very small organ. Okay, this is because as you age, thymus goes on becoming smaller and smaller and smaller. That is why it happens is something that you don't have in your syllabus right now, but you just need to remember that thymus is an organ that goes on becoming smaller and smaller as you age. By the time you're old, there's a pretty small amount of thymus that remains in your body. Now, what these T lymphocytes or T cells do is they leave your bone marrow and they start off their journey to go to this gland, which is called as thymus, which is located in your chest, right near this bone, which is called as sternum. Okay, you do not need to remember this name, but you need to remember the general location where your T lymphocytes are going to go. These T lymphocytes are going to stay over here for a certain amount of time till you have some kind of infection. And once this infection has started, these T lymphocytes and B lymphocytes will start to do a very peculiar kind of work. Okay, both your T lymphocytes and your B lymphocytes are going to be very, very efficient in killing different kinds of pathogens, and they are going to coordinate in a particular manner that they are very, very efficiently going to kill any particular kind of bacteria that might be infecting you. So now what I'm going to do is I'm going to show you the pathway in which these bacteria are going to be killed. Tell me if there is anything from this part about the lymphocyte that you want me to go over one more time. Anything that you thought, okay, maybe this one did not make much sense. The location is understood, where they go is understood.
Yeah.
Yeah. So you just need to remember where they are going to go, and depending on where they go, a very basic name is given to them. B cells or the B lymphocytes are called as B lymphocytes basically because they spend most of their time in their bone marrow, and later they go to your entire body. So remember, B going all over the body. When it comes to T lymphocytes, remember that they spend most of their time in the thymus, and therefore they're given the tagging T lymphocytes. That's basically the reason behind those different alphabets being used over here, right? Now, once we understand this, once we understand that, okay, this is exactly what these cells are going to do, there are certain other things that we want to learn now, okay? The most important thing that you want to remember about your B lymphocytes or the B cells is these cells have a very keen sense of identification of your plasma cells. Okay, plasma cells are all the cells in your body that are going to be able to secrete antibodies. So these cells are very, very efficient at identifying which cell is going to make antibodies. And along with that, your B lymphocytes also make memory cells. So I'm going to write this. These are two very important facts that you need to remember about your B lymphocytes, some memory cells. Okay, now what we are going to do is we are going to look at the mechanism of how B lymphocytes are going to carry out a function of defending your body. So there's a cascade, or you can say there's a pathway, okay, that we need to identify or we need to learn. Just give me 1 second. I have a doodle for this that I'll just pull up so that I will draw that particular structure only. It's not something similar to what you have in your textbook. This is a bit easier diagram to follow, and you can use this diagram to be drawn in your exam as well, and it just basically will still get you the same amount of marks. Just give me 1 second. I can't seem to see it anywhere. Okay, I don't seem to be able to find it. I'll just draw it out of my memory. Okay, so now let's say that you have this particular B lymphocyte here. Okay, now very important thing to remember about your B lymphocyte is that it already is going to have certain receptors on its body like this. Okay, what these receptors are going to allow your B lymphocytes to do is they allow the B lymphocyte to very specifically go and attach to certain cells that will help the B lymphocytes in initiating an immune response. Okay, now when we say that they help in initiating an immune response, what I mean is they are just basically going to allow your B cells, okay, to make a association so that those pathogens can be killed. So now I assume that this is a pathogen. The receptor on top of your B lymphocyte very specifically goes and attaches to that particular foreign pathogen, and it identifies that, okay, what I have with me over here is a pathogen. Now, once that foreign pathogen has been identified, this initiates a second response. So the first response is that it identifies the pathogen and goes and attaches to it. As soon as it goes and attaches to it, what it does, it starts secreting chemicals so as to begin the process of chemotaxis. Okay, using this chemotaxis, it brings in a very particular kind of cell into picture, which is called as T helper cell. And exactly like what the name suggests, your T helper cell is going to help the T cell to go and kill the pathogen. Right? That's literally in its name. What will the T helper cell do? The T helper cell will go and initiate a response so that the antigen can be killed. Now, this T receptor cell or the T helper cell is going to bring in your T cell, and the T cell will go and attach to this particular pathogen, engulf it and kill it. That is the basic function that is going to take place over here. Okay, but in order to make this reaction complete, in order for you to finish this entire work, okay, there are several smaller steps that need to be undertaken, which means like before you can eat your egg omelette, you need to crack the egg, you need to put it onto the pan, and then there's so many other things that you actually need to do before you can eat that omelette, right? Just having the eggs with you doesn't really help your case. That's exactly how it works with your B lymphocytes as well. So they are going to be doing this function, yes, but in order for them to finish that function, there are several paths, or there are several activities that they actually need to finish before they can get to that activity. So once they have finished this first part, okay, once you have your B lymphocytes that have identified this particular bacterium, they start to function as something called as the antigen presenting cell, or the presenter cells that we spoke of. So I'm just going to remove this part from here. I will write it in a pathway format so that you can remember it much more easily. So I'm just going to erase this portion away from here. Okay, so let's see. Okay, I have this particular bacteria here with me. This bacteria was just dancing around doing its normal shenanigans in your body, and all of a sudden you have this antigen presenting cell that comes into its picture. Okay, what is this antigen presenting cell? Nothing but your B cell. So I'm just gonna write over here, I have antigen presenting cell. What this antigen presenting cell is going to be doing, it is going to take this particular this particular bacterium, okay, and it is going to take this bacterium to your macrophage. Now there are two things that can happen, okay. If the bacterium are very large in number, okay, if there are too many bacteria present at this time, this antigen presenting cell can undertake the process of phagocytosis itself too. Mostly in order to make sure that these bacteriums do not move away somewhere, it is a very common practice that this antigen presenting cell will itself engulf this bacterium. So it undertakes the process of phagocytosis. Okay, who has undertaken the process of phagocytosis? APC. What is APC? APC is antigen presenting cell. Now, once this antigen presenting cell has engulfed this bacterium, so we'll make bacterium with a different structure like this. Okay, now once this bacterium has been engulfed, it secretes a vacuole around it like this, so that it does not interfere with the cellular activities of your antigen presenting cell. These antigen presenting cells, okay, are going to be identified. Okay, they are going to basically do a scanning process, and what they do is they're going to identify what particular kind of antibody will work against them best. So we are going to write antigens, okay, of the bacterium get placed on surface of the APC membrane. So you engulf this bacterium, you study this bacterium, and you find out what kind of antigen is present on its surface. So let's say you figured out that this is the type of antigen that is present inside this antigen. Okay, so you identify that this is something you need to see. These antigens get placed on the surface of your antigen presenting cell. This antigen presenting cell has just, in order to save your body, sacrificed itself, because as soon as these antigens are present on the surface of this cell, your immune system is going to identify this particular pathogen or this particular cell as a threat to its body. Okay, and then your activated B lymphocytes. So here I'm going to draw another cell, okay, so that I know this is where I need to take this. Now these antigens that are present over here, okay, they are going to be identified by your body. So these antibodies that have the similar binding sites are going to be attached to your B lymphocytes and they get destroyed. Okay, so now every single cell that has this antigen on its surface can be very easily identified by your body, because this B lymphocyte or your antigen presenting cell has gone and made itself similar to what the antigen is. It has made your bacterium a bigger target. Look at the size variations in the bacterium and this antigen presenting cell that you have. Because it has made it into a bigger target, what you can do is you can take this antibody from here. So let's say this is your antibody cell. Okay, so I'm just going to make it into a different format again so that you can identify that this is a different cell. Now it has these antibodies on its surface. Okay, these antibodies are very, very specific to the kind of antigen that is present on your APC. So now what happens is you will have this particular antibody containing cell. So we'll just bring it up and copy it and paste. Okay, somewhere here. Right now this cell is going to come closer over here, and what you are going to see happening in this portion is the identification and the matching up of this antigen and antibody, right? So this is what will take place. Now, once your antibody or the B cell is going to meet the specific antigen, so I'm going to write over here, the B cell meets the specific antigen, okay, on or inside the body, and it turns into a macrophage. So I'm going to write the antigen. Now, either this can turn into a macrophage and eat it, or you can say that it is just simply going to go ahead and kill these antibodies. Okay, there are different ways that can happen. For your body's efficient functioning, the most easier thing to do right now is to make these antigens and antibodies mix with each other. Once you have this process of antigen antibody attack that takes place, your body, okay, goes on to make multiple copies of this good B cell, because what it is able to do is it is able to neutralize this antigen that has come. So once you identify, okay, ye wala cell barabar hai, this is the cell that is going to work against this particular bacterium, you are going to give an approval to this cell, and multiple copies of this cell are going to be created. So I'm going to write multiple copies of this successful B lymphocyte get made. Okay, and out of these multiple copies that would be made, some of them go to kill those bacteriums in the higher number, whereas some of those cells will be turned. So I'm just going to make this here. Some of these cells will be turned into your memory cells, and they will be stored in your body, so that if this particular bacterium comes to you again, those memory cells will remember this process, and you will not have to do step 1, step 2, step 3, step 4, step 5, and then make multiple bodies in the sixth part. Next time, when the same bacterium infects you, the memory cells will only be activated. Okay, so I'm going to draw a little bit of brain on one of the cell. Okay, this turns into a memory cell. And next time when you have an attack, it is only the memory cell that gets produced. So we'll write gets stored in the body. And next time the same pathogen attacks, new cells will be made using memory cells. This makes the second infection last less duration. So if you get attacked by common cold for the first time, you will have the common cold for a very long time. But once you develop the antibodies for it, next time jab tumko sardi hoega, it will be cured much faster. So if first time it took you, let's say, about 11 days to get better, the second time you get this particular infection, you will have very less amount of time to recover from that. You will take only 4 days to get better, and that is all possible because of these memory cells. Is that making sense? Did you understand what I did over here? Yeah, I think
Yes.
you're on mute. Yeah, okay. So this is basically what is going to happen, so that your body can keep yourself very, very safer, and it can then basically make sure that next time you get infected, you're going to get better much quicker. Now, the T lymphocytes that we have, okay, they're also not going to be very different in their action. They are also going to make memory cells. The only part that you need to really look at when it comes to making these T cells is that the T cells have a bit of help with them. Okay, so they have something called as the T helper cell, and the T cells also need to be activated in the first place to do all of this. Just like how you have your B cells, the B cells are always roaming around in your body, and they are always going to be ready to take up the action. Like in case they feel like, oh, okay, so it's probably going to take a little longer for the macrophages to reach. So they themselves become like macrophages. Your T cells are a bit fussy, and they're like, nahi, hum aise nahi kar sakte. I have to follow a particular protocol, and I will use that protocol only to work. So let's look at the T lymphocytes ka action in more detail. So I'll write T lymphocyte. The T lymphocyte are always going to include two helpers with them. So I'm going to write include. First one is your T helper cells. And the second one that they are going to have is T killer cells. Okay, these are the two very important types of cells, and what sets them apart from each other is that both of them, okay, are going to have very specific receptors on their membrane. So I'm going to write both of these cells have a specific receptor on their surface. Okay, now what this receptor does is the key to their entire activity. What these receptors allow your T cells to do is they basically allow you to make multiple clones of themselves. So yahan pe bhi, when we said that you are going to make more cells, okay, there is still going to be a bit of structural difference in these all of the cells. They are going to have a similar kind of receptors on them, but the number of those receptors might vary, or you know, sometimes you may see that some of those receptors are not very specific to this particular kind of pathogen. There could be some or the other kind of differences. But when it comes to your T lymphocytes, the T lymphocytes very specifically make clones of themselves, which means that all of these cells that are going to be created are going to be exact identical cells of the same type, and they are also going to be created via the process of mitosis. Okay, now you are going to need to activate these cells in the first go so that they can work. So I'm going to write need activation. The activation could be by receiving a chemotaxis or because of the cytokines that are going to reach them. So when we said that you are going to have this chemotaxis that is going to take place, the kind of chemicals that are released are called as cytokines. Remember, the word kine means to move. Okay, so the chemicals that come out of the cell and move to other place are called as cytokines. They help you in initiating a chemotaxis response. Now, these chemicals called as cytokines are going to be secreted by your T cells, and these are basically going to induce your B cells to produce plasma cells. Now, I told you B lymphocytes can identify the plasma cells. They are going to make the plasma cells to secrete the antibodies. तो तुम्हारा जो T lymphocyte है वो पहले खुद activate होगा. Then it will release cytokines. These cytokines will reach, so I'm going to write it in very easy language over here. These cytokines go to B lymphocytes, induce the B lymphocytes to initiate response in whom? In the plasma cells to secrete antibodies. Okay, so at times what happens is your B lymphocytes, while they're roaming everywhere in the body, might fail to identify a particular pathogen. But T lymphocytes may have spotted them. And so T lymphocytes thoda sa घूम घाम के काम करके they tell your B lymphocytes that okay, देखो तुमसे ना ये point particular miss हो गया था. So here it is. Now what you have to do is you have to release the plasma cells. You have to tell the plasma cells to make these antibodies. And once these antibodies have been released, these antibodies will go and then they will kill the pathogens. Okay, so I'm gonna write the, इसको थोड़ा सा छोटा कर देते हैं. The monocytes or antibodies will destroy the pathogen. Okay, once this has already started, your activated T lymphocytes, so we are going to write over here, activated T lymphocytes will also have T helper cells. What these T helper cells will do, they will start to release certain cytokines like we spoke of. Okay, and what these cytokines will do is they will also take certain molecules to do the more functions. Okay, so they, instead of only releasing a particular kind of pathogen, they will go to another set of B lymphocytes and they'll tell them also to make more of your antibodies. Okay, once this process has also been initiated, then you will bring in your activated T killer cells. So I'm going to write about the killer cells first. Okay, now what is this activated killer cell? It is exactly what it sounds like. These are the cells that are going to go to the antigen, okay, and they are going to basically kill that particular antigen. For your activated T killer cells to function, you need a virus. So whenever you have a viral infection, activated T killer cells identify the virus and kill it. Okay, this is a very, very important point that you need to remember. Once they have identified, once they have killed that particular virus, some of the T killer cells, turn into memory cells, and these memory cells get stored so that the next infection can be cured quickly. And that is all that you need to understand for the pathway in which this is going to be done. When we talk about the T killer cells, they are going to digest the virus away. Okay, so I'll just do this.
The chapter hasn't ended yet.
No, it hasn't. It has antibodies and it has vaccination and that has vaccination program. That is all the questions that you are going to get the data for. Okay, तुम उसको बस पढ़ लोगे तो भी चल जाता है, because in your vaccination, the only part that you need to remember is that you are going to have different ways in which vaccine can be produced. The one way is where you can get infected by the pathogen and then your body will remember it and then it will do all of that fighting for itself. Or sometimes your doctors will take an initiative and they'll make the vaccine outside. So how they make the vaccine outside is they take some bacteria and they give those pathogens inside the body of your bacteria and they infect the bacterium, and then the bacterium make antibodies and we collect those antibodies and then we give them to you. If we want like more specific kind of antibodies, we'll give those pathogens to a mouse and then we'll tell the mouse कि like भाई देखो तुम antibody बनाओ. तो वो mouse के अंदर एक particular मास ऑफ सेल्स बनता है जिसके पास एंटीबॉडीज होती है. हम उस मास ऑफ सेल्स को आइसोलेट करते हैं, फिर उनको टेस्ट ट्यूब में ग्रो करते हैं, एंड देन वी किल दोज सेल्स and we extract the antibody out of them and we give it to people. The problem with making antibodies like that is they're very specific. They're one particular kind of antibody, and those antibodies are called as monoclonal antibodies. Mono because one, clonal because it's made from the same type of cells that we clone in a test tube, and they're very specific because they're one type only. So at a particular point of time तुम mass production कर सकते हो एक particular disease के लिए vaccine का, like what we did for corona right now. We made one vaccine and we started to give it to people, and by the time entire world was vaccinated, the virus mutated, and we were like, oh damn, we made antibodies only of the first type, so अब हमको second type की antibodies वापस बनानी पड़ेंगी. And then the scientists started to do that, and तब तक virus वापस mutate हो गया, and then people were like, oh damn, we made antibodies of the first type only, we'll have to do that again. That is the issue with making vaccines of one type, and therefore monoclonal antibodies are not very famous. Okay, what we are doing right now is we are relying on monoclonal antibodies, and it's failing really bad. But what can you do? Government decided to make monoclonal antibodies. Okay, so that's what you need to remember. Yeah.
Yeah. So, is there a chance that a body can destroy, like a virus or something?
Yes, you can destroy the virus, but the point is, by the time you get to destroy, by the time you identify and destroy the virus, sometimes what happens is the virus kind of changes its structure. This happens with most of your retroviruses, like the virus for your HIV, and apparently also your coronavirus. So what happens with these viruses, okay, is mostly they have a specific structure when they enter your body, and it is quite possible that by the time they entered your system already had an antibody to kill them, and it will kill that particular virus for which the antibody was present. And during that entire encounter, your body misses that other virus which also entered with it but was mutated. And because this was a mutated virus, तुम्हारी body के पास इसको specific antibody नहीं था. So your body will kill the first type and the second type will escape away because you did not have this matching antigen for it, antibody for it. And so it becomes very difficult for us to cure any viral infection. And so we just normally say viral infections are very common. In fact, we also say that cold is very common. Cold is caused because of virus. So there are different types of viruses that give you cold, but the common symptoms are that you get fever, then you get body pain, and then you have like this uneasy feeling for some days, and then it gets better. And so we call it as common cold. And some people just don't get common cold because their body can kill the virus, but it's not very common thing to kill the virus. Your body needs a lot of efforts to do that. Right? That makes sense?
Yeah, okay.
Okay. What else? Apart from this, there's literally nothing else that you need to know in this chapter. There will be a lot of data that will be provided in the questions, and all that in this chapter you need to do is you need to just read that particular data and understand that, okay, if A happens, then we have to do B. There is one more concept that you have learned previously, and I'll just go over it one more time, which is that as default, okay, babies are born without an immune system. So they don't have any immune system of their own for the first 6 months of their life. So for the first 6 months of their life, they get the immunity from the mother, right? And because you're getting this immunity from the mother, we say that this is something that is given to you. Okay, so I'm going to write the word given. And then by the time you become 6 months, certain amount of cells that you had in your body start to mature, and then you can defend yourself. So the immune system that you are born with but is not activated throughout your life, it gets activated once you become a certain age, is called as innate immunity. Something that was present in your body, but it wasn't activated for the first 6 months. What you get from your mother is the acquired immunity. Okay, you acquire it because your mother gave it to you. You can also get acquired immunity by taking certain kinds of medications, or you can also acquire immunity by getting infected with a particular pathogen. This is something that you did not have in your body, but you got it or you acquired it because you did something to get that infection. When you get the antibodies from your mother, but you were not infected with anything, right? Your mother just gave those antibodies to you so that you don't fall ill. So you're not really doing anything. That antibodies or those number of cells that you are getting from your mother are not making you sick, they're making you stronger. And therefore, because this immune system is not really functional, it stays inactivated inside you, we say that the kind of um, immunity that you get from drinking your mother's milk is passive immunity. Why passive? Because you get the immunity, but your body doesn't really do anything with it. It doesn't make you fall sick. It doesn't make you have cough. Nothing. You just get it. But let's say as a child who is 6 or 7 months old, you get infected by a particular pathogen, and then your body, along with certain vaccines that people will give you, if you have the ability to defend that infection, then the immunity that you get because of the vaccine is called as artificial immunity. Why? Because this immunity was not made by your body or was not given to you by your mother, but it was artificially injected inside you, and therefore it is called as artificial immunity. Artificial immunity or vaccines are not made inside human bodies, they are prepared in labs, and that is very important that you understand. So the immunity that you get from your mother, or because you fought that particular infection off, will be called as natural immunity. And the one that you get because of medication or because of vaccines will be called as artificial immune or the artificial immunity. You can also get artificial immunity when you do organ transplant. So let's say you got an organ like kidney from a particular person who was infected with a very deadly disease, like let's say they were infected with malaria or they were infected with tuberculosis. Usually these people are not allowed to donate their organs, but let's say okay, they got like this at a very young age and they donated their organ when they were 40. So all those antibodies will still remain in them, and you just kind of get those antibodies in you because you got their organ. Because it was in a live body, it will still be called as natural immunity and not artificial immunity. But if you get an organ, and because your doctors were suspicious that तुम इस organ को लेने के बाद मर जाओगे, so they give you certain medications to keep you safe, then it will become artificial immunity. So something that is chemically made and then injected in your body is always artificial immunity. Making sense? That's
Okay.
all that you need to know. I'm going to share with you a YouTube link for that monoclonal antibody. All that you need to do is you need to just look at the steps. They have not ever asked to tell you the steps of preparation of a monoclonal antibody, but it helps if you're prepared for all the things. So I'm just going to share that YouTube link with you. I just want you to see it and maybe write down all the steps that you see and then send them to me. And if you've missed anything, I'll just correct that part and I'll send it to you. And I'll also send this small notes wala section that we prepared so you can go over this as well. Okay, so I'll make this a little slower and I'll share it with you and you can keep it for your recap. So just later on, just take a look at all of the points that I have written down and maybe read the chapter ones. That really helps. So those are just the points that you need to know for this particular part. All right.
Yeah.